Okay, hi everybody. So I'm learning new things still. Sorry for this. It's not ideal, I know, and this is why we don't record lectures all the time and post videos on Moodle because it's better to be in person. But this was what has to happen today. So a couple things before we start. Make sure that you have observed your plates from lab last week. You have the mannitol salt plates and the pea-glow plates. Remember, if your pea-glow plates didn't work, you need to look at somebody's that did, and I left at least one group back by the UV light that did work. Once you have observed plates, unless yours is a group that worked and is sitting back in the back, you can put your plates in the biohazard waste. Lab this week will be meeting in our normal room, which is 214, and we'll talk about that. Also, just looking through everything and thinking about it, I decided I think it would be better to have the test on Monday instead of on Friday. So the test will be Monday, so that will give us this week. We're going to finish chapter 27 today and start chapter 28. We'll have time to cover things in chapter 28, and then on Friday we'll finish chapter 28 and then do a little bit of review for the test. So the test will be Monday. That'll change a couple things on our syllabus, but it's not a huge deal, and I think it'll be a better solution. So you see on the screen, hopefully, I think you see on the screen, some questions from Friday, so you can just take a minute to think about those. I would actually just say, why don't you pause the video, because I'm just going to keep going. I could keep going forever and ever and ever, so I will stop at 50 minutes, just because that's what our normal lecture time is. So, pause your video, take a minute to try to answer these questions, and then I will give you the answers. Okay, so one difference between gram-positive and gram-negative cell structure. You can think about the thickness of the peptidoglycan layer. You should remember that gram-positive have the really thick one and gram-negative have the thin one, and that affects how they take up the stains for gram staining. Also, the gram-negative bacteria have that outer lipopolysaccharide layer that the gram-positive bacteria don't, and that allows them to be more resistant to antibiotics and more resistant to the immune system. And sorry, I have a bird clock. And Thomas is playing in the background. Number 2, which bacterial structure or structures helps a bacteria adhere to a host or substrate? There's 2 answers here, Fimbriae and the glycocalyx. You should remember what all of those other structures do as well. The pili is for conjugation. It's longer, but it looks structurally the same as a Fimbriae, but has a different function. Fimbriae are smaller. Can be a lot of them, and they're used for adherence to a host cell or to a surface. The glycocalyx can be organized or unorganized. So if it's organized, it's a capsule. If it's unorganized and looser, it's a slime layer. Endospores are to just help protect a cell. If conditions go downhill, they can store their genetic material in the endospore until conditions are good, and then they can germinate to be a vegetative cell again. And flagella are just for movement. And 3 ways a bacteria can undergo recombination. Transformation, they can take up external DNA like a plasmid. Transduction, a bacteriophage can transfer DNA in between 2 different bacterial cells, or conjugation, where 2 different one bacteria can give DNA to another bacteria through the pilus, which we talked about in the last question. Okay, so we kind of left off, and we need to talk a little bit about some of the differences between archaea and bacteria. We're not talking a huge amount about the archaea domain, but here's a chart that compares and contrasts the eukaryotic cells, eukarya, the prokaryotic bacteria, and the prokaryotic archaea cells. So you can see it talks about nuclear envelope, and because you know that both bacteria and archaea are prokaryotic, that neither of them have a nuclear envelope, and eukaryotic cells do, again, because they're prokaryotic. Sorry, trying to get my technology in order here. Neither bacteria or archaea have membrane-bound organelles. Their cell walls are made of peptidoglycan. If they're archaebacteria, their cell walls are made of something that's similar to peptidoglycan, but not exactly the same as peptidoglycan. And eukaryotic cells have them. Some of them have them, some of them don't have them, like animal cells do not, but plant cells and fungal cells do. The membrane lipids found in the membranes, some are branched, some are unbranched. Bacteria, you'll see bacteria and eukarya are similar here. Antibiotics, bacteria are similar sensitive to antibiotics, but the other two are not. And then the DNA structure is variable. So bacteria have singular circular chromosomes just like archaea, but they can take in plasmids and they don't have histones, whereas archaea might have histones. And eukarya have multiple linear strands of DNA chromosomes and histones and complex packing, which we've talked about. Other things to think about with the archaea is just that they tend to be extremophiles. So extremophiles meaning they can live in crazy conditions. Halophiles can live in high salt conditions, which we talked about at the beginning of the chapter with the Great Salt Lake. Thermophiles live in really high temperatures like the hot springs. There's some that are what we call methanogens. So they actually live in a normal temperate environment, but they release methane as a byproduct. If you've ever been to a swamp and it smells like rotten eggs, that's because of these prokaryotic cells. They are anaerobic bacteria if they're methanogens. They're anaerobic, so they don't use oxygen, and methane is a byproduct. They can live the methanogens can live also in Antarctic ice. So there's a couple different kingdoms within that, but you don't need to worry about learning all the names of them. But a lot of the kingdoms, divisions in the archaea are based on where they live or the conditions that they can survive. So most of the halophiles are in one kingdom, most of the thermophiles are in another, but it's not totally true because classification is fun. So again, just knowing that overview and thinking about the fact that they're extremophiles is what you need to know about archaea. Okay, whoops, sorry. And here's where we want to talk about the fact that bacteria are not all bad. I might have mentioned that before because I feel kind of strongly about that. They have a lot of different roles that are important in the biosphere, and most of them are not harmful. We just tend to focus on the harmful ones, which we'll do in the Protist kingdom too. So they're critical to the survival of all life on Earth, and they have a lot of different roles. Nope, sorry. There we go. They're used in making food. Some foods that you might enjoy are somewhat dependent on the presence of bacteria. Things like fermented drinks, maybe you shouldn't enjoy those so much. Cheese, soy sauce, yogurt, kimchi, kombucha, I don't know how you enjoy that, but some people do. Pickles, vinegar, sauerkraut, all those kinds of things have bacteria involved in the production. They're also really useful in research, and you kind of saw how we used E. coli in the transformation experiment, but just understanding processes like that at a bacterial level can help us also maybe scale it up to understand or do research with eukaryotic cells. Chemical recycling, again, really can't overstate the importance of how important they are in recycling chemicals. They're decomposers, so they break down dead things, and they release nutrients that are trapped in the dead bodies to be used into the environment again. So they break down dead organisms. We use them in the process of cleaning up sewage. Bacteria are really important in digesting the organic materials that are found in sewage. They free carbon up, which is important for so many things, and nitrogen to be reused, sulfur, phosphorus, all those kinds of things. They're also fixers, which we've already kind of hinted at. They fix carbon through photosynthesis. So they take carbon out of the atmosphere where it's not in an accessible form for heterotrophs like us, and they fix it by putting it into an organic molecule like a sugar. They also do that for nitrogen. We talked about that on Friday, I think, that there's a lot of nitrogen in the atmosphere, but it's not accessible. I'm just going to write carbon and nitrogen there because those are the big two important ones. They're bacteria that live in the root nodules, especially of plants called legumes, and they take nitrogen out of the air and put it into the soil to make it useful, and we've kind of harvested that process and made it more inorganic, but to make fertilizers as well. They also make molecular oxygen. Cyanobacteria are really important. The photosynthetic process of cyanobacteria is really important for releasing oxygen into the atmosphere, which we all appreciate. They're also involved in a lot of symbiotic relationships. So symbiotic relationships involve two organisms, and they're different forms of this. So the two organisms live in close contact. There's a host and then there's a symbiont, which is the organism that is living in close contact. And there can be various forms of this relationship. So in a mutualistic relationship, in mutualism, both the host and the symbiont benefit in some way. So you see in this picture, this is the intestines, and you've got all these little green and blue organisms. Those are bacteria. This is, for the most part, a mutualistic relationship. So we have lots of bacteria in our intestines. They help us to digest food, to get nutrients that we couldn't get other ways. So again, with that chemical recycling, but they also make vitamin K for us. E. coli is really important, making vitamin K. And they benefit because they get a lot of food. And so they're still getting food, but we're also getting a benefit from it. So both the host, us, and the symbiont, the bacteria that are in our intestines, the gut microbes, are benefiting. In commensalism, one organism benefits and the other one doesn't care. So they're not helped or harmed, and usually it's the host is not helped or harmed and the symbiont is benefiting. So an example of this is skin bacteria. You have lots of bacteria that live on your skin, as you may have seen when you did your nose swab. But generally the skin bacteria are benefiting from you and you're not really getting a benefit from them. There's some research out there that suggests that there aren't really any commensal relationships that really it goes one way or the other, but we still categorize some relationships as commensal because we don't necessarily fully understand them. Skin bacteria could be helping to keep pathogenic bacteria from growing on your skin, or so we could be getting a benefit from it, but we define it right now as a relationship where the bacteria are benefiting, the symbionts are benefiting, and we are not helped nor harmed. And parasitism, obviously, one benefits, that's the symbiont, and one of them does not benefit, and that's the host. So the host has negative consequences because the parasite is just taking from them and the parasite gets stuff. And so these are things. We also talk about this in terms of pathogens, which are bacteria, parasitic bacteria that cause a disease. I'm trying to think. Other things that bacteria do for us, they help make antibiotics. So a lot of antibiotics that we've discovered are compounds that are produced by bacteria, and those bacteria are producing those compounds to reduce competition for themselves, but then we get the benefit of that. They make vitamins, as we talked about, in the intestines. They're also involved in bioremediation, which is cleaning up like oil spills. There are some bacteria that will actually break down petroleum, and so that's called bioremediation. So we can use bacteria to help clean up our issues, and they're actually used in the process of making biodegradable plastic. So they have lots and lots of roles, and again, we're continuing to understand more and more about that, especially the mutualistic relationships that we have with our bacteria that we don't even know about. We're continuing to understand those more and more and more. Okay, last slide of chapter 27. This is a list of a whole bunch of pathogenic bacteria. So pathogenic bacteria, we're not going to go through all these, but what I would think what you might want to do if you're studying this, say for a test next week, you need to think about what do we need to know about a bacteria. When we talk about a pathogenic bacteria, one of the first things we always want to know is what we did in lab. Whoa, that's a funny G. Is it gram-positive or gram-negative? So looking at that helps us to understand how to treat it, because remember, gram-positive are more susceptible to antibiotics and gram-negative are not. You can also look up, you don't need to do this necessarily for all of them, but let's say pick 5 and research them. What are their symptoms? Are they gram-negative or gram-positive, and what do we do to treat it? So in class, what I was going to do was actually encourage you to pull out your devices, which I never like to do, but it can be useful, and pick one to research, and then we were going to talk about them quickly together, but I'm not going to go through and list this all off for you because it will just be horribly boring for you as well. So you pick 5, and I'm going to say that might be part of your quiz that you're going to take on this chapter. So pick 5 and look up, are they gram-positive or gram-negative? What are the symptoms that they cause, and what is the treatment for them? And that is going to wrap up chapter 27 for us. So that's bacteria. your outline for that. I changed the due date to be due on Wednesday because we hadn't finished talking about it in class. Okay, give me a moment. I'm going to just pause this for a second, maybe. Okay, so this is chapter 28. So chapter 28, we're going into, we've covered 2 domains. We've talked about the domain bacteria and the domain archaea. We're heading into the domain eukarya, and that's where we're going to spend the rest of the semester. So the domain eukarya is all eukaryotic cells, and we're starting with protists, which are possibly like the weirdest random classification. So we talked about the whole idea of like domains, kingdoms, phylums, class, order, all of that kind of stuff. And then we're going to start with protists, which aren't really any of those things. So protists used to be a kingdom, but they are not anymore because we've discovered more and understand more genetically where things come from and that kind of thing. They're still kind of, it's still kind of a weird category, and people talk about protists like it's a kingdom, but it's not a kingdom. So we're moving into eukaryotes. Protists is what we call an informal grouping, and basically what it is, these are eukaryotic cells that don't fit into the other 3 kingdoms, which are plants, animals, and fungus. So these are, I always called this like the leftover kingdom, so now it's not even a leftover kingdom, it's like the leftovers. We don't know quite what to do with them. They don't have a lot of unifying characteristics. They don't have a lot of things that they have totally in common. They're small. Why is it not doing this for me now? Okay. For the most part, they're usually unicellular. So they're mostly one-celled, but that we'll talk about today even is not always true. A lot of them are aquatic. So most of them live in water, freshwater and saltwater both. And some people will talk about, like, we're gonna use the term supergroup. Some people will say that those are their own kingdom. So we don't even really necessarily agree on their classification. So in this picture, what you see, and you've got a scale here of 1 micrometer. All of these are protists except for 1. So you guys are probably too young, but there used to be a Sesame Street song that said like which of these is not like the other. So again, if we were in class, I'd say which one of these is not like the other, and those of you who've read chapter 28 would know, and those of you who haven't would just have to guess. It's this one. That's the only one that's a prokaryote. All the rest of them are protists, so they're eukaryotic cells, and you can see they look pretty similar, and it would be hard to distinguish that just based on looks. So we're going to talk about very specific characteristics to help us put things into categories, and still just keeping in mind that this is still kind of a random, informal grouping of things that we don't know what else to do with. Okay, so again, we talked about that protists are in the domain eukarya. They are not a kingdom. They are just an organization. There's a really good overview in your book, which I failed to look at the page numbers for. I think it's 594 and 595, that has an overview of the kind of 4 groups that we're going to be talking about. And you can see these here. We're going to go through these 4 groups in pretty specific details. You need to be able to compare and contrast the groups, so knowing what's similar and what's different, and you also need to be able to just know the specific organisms that we talked about and how they're classified and why they're classified that way. Again, these classifications are changing, so like I'm looking at this diagram, and this diagram is different than the edition. Like I have the 9th edition of the book, and you guys I think have the 10th or 11th edition of the book, and my diagram in my 9th edition is different because this keeps changing, and you'll see things like dotted lines in these, you'll see things like dotted lines in these phylogenetic trees. That's because things are changing, we're not certain of things, and again, this is supposed to be showing common ancestors. This shows like one common ancestor here for all the protists, and we don't necessarily know that that's true. They could be polyphyletic instead of monophyletic. Again, what do they have in common? They're eukaryotic, so they have a nucleus. They have membrane-bound organelles. Most of them are unicellular. Lots of them are aquatic. Um, this is a very diverse kingdom in structure and function. Not kingdom. I keep saying kingdom. I'm sorry. It used to be a kingdom. Some of them have very unique organelles, which we'll talk about, and that's some of the reasons that we put them into these 4 different groups, is based on those unique organelles that they have. Some of them are autotrophic, meaning they make their own food. Some of them are heterotrophs, meaning they consume food. Some of them are mixotrophs, meaning they can do photosynthesis, but they can also consume other nutrients. They reproduce asexually. They can reproduce sexually, some of them, and they have all different kinds of variations within that. So again, not a lot of common things that tie them all together. So we're going to be looking at what makes these 4 groups unique, and why are we classifying organisms into these 4 groups. So there's a lot of weird names, a lot of new terms, a lot of vocabulary. I would recommend making flashcards and thinking about that kind of thing, so that you're practicing knowing these names and what organisms belong in which group. I'm sorry, in addition to children, my cat also wants to be involved in this lecture. Okay, so we're just going to kind of dive in. I'm anticipating we'll get through a little bit of this. This supergroup and part of SAR is what I'm thinking. In lab this week, this is what we're doing. So we're going to be looking at these organisms and seeing them in real life. So not just looking at pictures of them. So this is relevant to what we are doing in lab. This is exactly where we need to be. Okay, so Supergroup Excavata. So again, we're using the term supergroup. This is kind of an organization that doesn't fit into kingdom, phylum, class, order, family, genus, species. It's just a group of organisms that are in the Protists informal group that we are calling Excavata. They're called Excavata. So they do have some things in common. One of the things, they all have, this is failing on me too. They have a feeding groove. Oh my word, I can't write anything with this is dying on me. I'm going to just have to use my finger. So they have a feeding groove that says feeding groove, which is excavated on their sides. So they also have a unique cytoskeleton. They're put into this group based on the classification, the characteristics of their cytoskeleton. We're going to talk about 3 different groups. There's 2 on this slide, and we'll talk about 1 more. 2 of the groups that we're talking about, and a lot of the groups that we talk about in general, in Protists have... Oh, that's not going to work either. Wow. Oh, I can't write with my finger either. That says nothing that makes any sense. We're just going to erase it. They have modified mitochondria. I'm just seeing if I can type that in there, but I can't. So 2 of the groups have modified mitochondria, and others have flagella that are structurally different. So those are things that are helping us categorize these organisms this way. So we're going to start with the diplomonads. Diplomonads. I think you can say it either way. So these are organisms, and that's what's pictured here. And I'm trying, still trying to write things with my finger now, and it's not working. That's what's pictured here. They are unique. We talked about having modified mitochondria. They have modified mitochondria, and their modified mitochondria are called mitosomes. Mitosomes are unique. You may remember talking about mitochondria having the electron transport chain. Mitosomes don't have an electron transport chain that functions. So no electron transport chain means they can't use oxygen. So these bacteria are anaerobic. Also, their mitochondria have no DNA. So like you'll hear talk about mitochondrial DNA. Some mitochondria actually have DNA, and these do not have DNA, and the DNA that's in the mitochondria is different than the DNA that's in the nucleus. So these don't have any DNA at all, but they do have 2 membranes. So they're modified mitochondria called mitosomes. Many of these have flagella, lots of flagella, and they all have 2 nuclei. So you can see they look like they have eyes on them. These are both Giardia intestinalis, The You can see the two nuclei are the same size, and they've got these flagella floating around. The flagella, remember, are different than prokaryotic flagella. Um, they are made up of flagellin, is the protein, and these are attached to the cell surface, so they're not coming out of the cytoplasm. Um, and I'm trying to think what else I was going to say about that. I feel like there was something else. Sorry. No. Okay, so Giardia is found in untreated water. So this is like a camping kind of thing. Like if you're drinking water out of a stream, Giardia is something you would be at risk for. It causes intestinal issues. And so you should boil water, or you can add iodine to water if you're drinking water out of a freshwater source. You're at risk for Giardia. Okay, so that's an excavata, that's a diplomonad. A specific example that you need to know is Giardia intestinalis. Okay, the second group is the parabasalids. I told you there's a lot of weird words here. Parabasalids are also excavata, so they have that feeding groove. They also have modified. Why is my finger sometimes working? Sometimes doesn't. They also have modified mitochondria, and their mitochondria are called oh my goodness, this is ridiculous. This is going to take more time than it's worth. They're called hydrogenosomes. Hydrogenosomes, they have hydrogen in their name. That's because they can produce energy anaerobically, and when they do that, hydrogen is a byproduct. So anaerobic respiration with hydrogen gas as a byproduct. Again, no DNA, but they are double membrane, so they're similar to the diplomonads. Again, I said we tend to focus on the bad ones. We're focusing on the bad ones here. The example that we're talking about here. Oops. Oh, there's the word. Oh, good, Rachel. See, I thought I split up this slide. I'm just talking to myself now. So the example that we talk about here is Trichomonas vaginalis, which is a parasite that obviously can affect the vagina, the vaginal area. It's an STD. It causes trichomoniasis. They have flagella, and they have an undulating membrane and that's what lets them travel up the vagina. It actually can affect male urethras as well, but it's usually asymptomatic for males, whereas it's not for females. For females, it really only usually becomes a problem if something changes with the pH of the vagina, and that can make the symptoms appear. So we will be looking at Trichomonas in lab. I don't think we have a slide of Giardia. We don't have live of either of those. Okay, one more slide, I think, and then we're going to be done for the day. If I can make this do what I want it to do. Okay, so the last group of excavata that we're going to talk about today are the euglenozoans. Again, practice these words. I don't care if you know how to pronounce them. You need to have a reasonable idea of how to spell them, and you probably need to be able to recognize them for sure. And again, we'll be looking at these also in lab. So euglenozoans are they are protists that have a unique flagella. So their flagella either has a spiral or a crystalline rod inside it. So they're flagellated, and we divide them into two groups, the kinetoplastids and the euglenids. The kinetoplastids have a kinetoplastid. So a plastid is just an organelle that it's a membrane-bound organelle. So the kinetoplastid is a really large mitochondria. They have one really big mitochondria that has a pile of DNA in it. Most of these are heterotrophs or parasites, so they're getting their nutrients from other things, and they're parasitical. So again, we're focusing on the bad. There's two kind of trypanosomas that are problematic, cause two kind of famous diseases. One is African sleeping sickness, which is caused by a trypanosome that is carried by the tsetse fly. So it's common in parts of rural Africa, and it causes things like fever, headache, irritability, fatigue, swollen lymph nodes, aching muscles, typical kind of disease things, but it can progress into neurological symptoms like confusion, personality changes, if it gets into the central nervous system. So that's caused by a trypanosome. The other one is Chagas disease, and Chagas disease is more common in the Western Hemisphere because it's found in um, a specific kind of insect called the triatomine bug that's found in Central and South America and all the way up to Mexico even. It's a blood-sucking insect, and it's commonly pretty mild, but it can progress to the point where it causes congestive heart failure. So it can be pretty serious. I thought I had a number written down about how much this happens. Where's my number? I guess I didn't write down. Oh, there it is. There's about 200,000 cases a year of Chagas disease in the United States. So again, both of those are caused by trypanosomes. We'll look at a slide of that. What's hard about these is they can actually switch their surface proteins. So they have surface proteins on the outside that identify them as, hey, I'm a trypanosome. But every generation they switch those. So they're hard to treat because they're hard for your body to be able to react to because they keep switching surface proteins. So that's a strategy that they use to avoid detection and attack by the hosts, is they have regularly switch their surface proteins. On the other hand, on the other group of euglenozoans that we talk about is the euglenids, which seem really nice compared to the trypanosomes, compared to the kinetoplastids. They have a pocket at one end, which you can kind of see over here. They have this little pocket, and that's what the flagella comes out of. And they usually have two flagella. This one's not really showing that. Some are mixotrophs. Most of them are mixotrophs. So they are able to use photosynthesis, so you'll see they're actually green. But they can move around, and if they can't get to light, then they're able to actually absorb nutrients or phagocytize, like absorb through diffusion or phagocytize other things to be able to get their nutrition. We will look at these living in lab, and they're just cute. They just zip around, and they're green. And sometimes, if we're lucky, you can see this red eye spot, which is here and here. So they have an eye spot, which actually helps them. It actually acts as a shield to try to get the right angle of light, and then behind it there's a light detector, and so they can actually move toward light sources to get light, because then they can do photosynthesis. Again, they're just really cute, and I like to watch them. I'm trying to see where we are time-wise. Let me just check. Okay, we are just going to stop there. I think we're a little bit short. Actually, I know we're short, but the next pile of stuff is just really long, and I don't necessarily want to get into it. So, after you've watched this video, there will be a quiz on Moodle for you to take. It's a short quiz, but that's kind of a way for me to make sure that you've actually watched this video. Okay, so watch this video, and on Wednesday we're going to pick up with the Supergroup SAR, and we'll probably get through SAR on Wednesday, and maybe a little bit I don't know, we'll see how far we get on Wednesday. So, watch this video, take the quiz by midnight tonight, and I will see you on Wednesday.